It's my pleasure to introduce the first speaker today, Veronica Grieneisen. She was at John Innes Center, but now she's at Cardiff University. Veronica.
Thank you. So it's this one, right? OK. So welcome, everybody. And it's really a pleasure to be here. So I will be talking about polarity. So this is the general theme. But you're going to see a couple of things. You're going to see a lot of plants also, and also some animal cells. But actually, I would really want to lift this talk conceptually. So in the end, you're not going to think, OK, this is about that paradigm system or about that organism. But really, it's about a process, OK? And I hope I can, in the end, link it back to many of the themes we've been seeing passing by yesterday. So, well, the way I see this meeting is that we are trying to understand how we can get transformation, so morphogenesis, right? Either multicellularity from one cell to a complex organism. But it's also like this, this picture, this painting done in the 30s. It shows a little bit this, like the absurdity of what we're trying to do here. The task of going from genetics to anatomy, right? And there's a huge sort of magical jump there. And how is this happening? And I don't think I have to tell you guys, you're convinced that just by looking at a sequence of the DNA, for example, we're not going to be able to do such a jump, right? Something is happening there. Some processes are happening. So in the lab, we're very much interested in the body plan of plants. So this is from Julius Sachs. And I want to explain to you, actually, that this is a very interesting thing to look at. And there might be hidden messages in this system by understanding it, which could be applicable to other systems. And it is actually very interesting because some people are fascinated as a biologist, like, oh, how would life look like if it had been evolved again somewhere else, if it weren't this life? And in a certain way, we have the luxury of asking this question here because we have multicellularity, which has arisen, as we heard yesterday, at least 20 to 30 times, right? So in a certain way, if you think about ourselves and a plant, it is pretty much an alien. But it's a much more interesting alien than this anthropomorphic one here that we find cute because it has eyes. And that's because this one here will always have, like, it has 2 legs, 2 arms, a certain number of digits. So we like that. But you have to now understand that plants, they're very, well, these are different species. So you'll see they have different strategies in their body plan, which is really to make them adaptable to their environment, right? So this has happened, this adaptation over longer time. So through the gene regulatory networks, plant-soil interactions, environmental conditions, and ecological interactions. But more importantly, even if you take the same genotype and you grow this, so these are Arabidopsis plants, and you grow them under different nutrient amounts. So nitrogen is something which is really important for them. So these are all amounts which would be OK. So physiologically, they're sort of happy. But what you see in low amounts, they have to forage. So they have to make more lateral roots. So they have a higher density of lateral roots, and they'll be longer than if you give them more food. So if you give them more food, they're happy and they become lazy, right? So what you see is that the body plan, the number of lateral roots and the length and the growth rate, these things are changing because of the environment. So that makes it more interesting than just thinking, OK, what is the pattern which is robust and which is always being displayed? So below this, and I'm not going to go into this, is not the point of the talk. It's really just to prime up the motivation here of my second part. So below this here, we know that's a lot of the receptors, trans receptors for nitrogen, for nitrate. And downstream, there are a lot of targets, genes, and they will promote primary root length, lateral root density, and so forth, right? And this is in a dose-dependent fashion. OK, that's all very nice. However, we have a problem. And that's when, for example, you grow the same plant, right? And now you offer, for example, this is a low global condition, but you offer a little patch of high nutrients, OK? Now, although these receptors are along the axis of, right, on the epidermis of the plant, what happens is exactly the opposite reaction will occur. So now where it is high, because that high patch is localized and the rest of the environment is at low food amounts, it will very intelligently only promote the growth at the region where you have the high amount of food. In this case, though, it had stunted it. It had stopped, right? Because it was offered globally. And the same thing happens here. So what you can see, the plants, they will change the growth rates, but also the density, so the number of meristems it'll make. Okay, so this is an inverted behavior, and it's quite paradoxical if you would think about it only about the cells at a location feeling its immediate neighborhood. So this is very similar to what you saw, for example, when Michael Evan was showing you about the like cutting, right? That now there has to be communication between local parts and global parts. Okay, so this communication not only is for patches, and these are experiments we're performing with von Vieren in Germany, but also, for example, if you now split the root on top, so they're totally two independent roots and they only meet at the shoot, but they will know what the other side is sensing, okay? So there's this global communication also between the organs. So what is this moving signal, right? What is this thing? And the idea that plants have information which is moving along them very quickly is something which Charles and his son, Francis, they were very intrigued about, and they wrote big books, right? One called The Power of Movement of Plants. And in it, although they didn't know what it was, they already could say that there was something being transported polarly, right? There was this movement. What that is auxin. So there are many things actually moving around, but auxin is a very important signal for plant development. And I don't have to go too much into it because you heard a lot about it yesterday by Elliot. But just to say, just to remind you again, so this is again just this picture that Elliot explained yesterday about the chemiosmotic theory, right? So auxin, it can basically permeate into the cell membrane, and once it's in the cell of a plant cell, it gets trapped. And it needs something, right, to put it back out there. And those things, are the pins. So these are these proteins which will efflux auxin. And it effluxes it not into the next cell, but into the cell wall, and there it can be picked up and so forth. Okay, so we have this indirect type of route. But if you then look, for example, and this is the tip of a root of Arabidopsis, and you look at where they are positioned, all of these proteins, which are these pumps, and you look at their polar location for every cell file. And this is something we did, like this is the first thing I did when I thought about plants ever in my life. You can make a model where you sort of make a caricature of the root, and you allow the auxin to enter the root because it's coming from the shoot, right, from the leaves. And what you see is just because of the distribution of these pins, right, which here in the central files are located downwards, these flanking files here, these blue ones, they're located upwards. And here, these yellow ones, they're a little bit inwards. And here on all the sides. You get an inverted fountain, a flux, and that will generate an exponential gradient of auxin. And that, in fact, we think is acting as a signal for a morphogen for this meristem to grow, to divide, and also to know when to differentiate. The interesting thing about this morphogen gradient here is that this is an example of a morphogen gradient which is extremely robust, which can grow with it can follow the growth of the root because of the transport rates being very high. It does not depend, though, on diffusion. On the diffusion rate. So the diffusion rates will not change the characteristic length of this gradient. So we have a different control mechanism here. So if you think about Arthur Lander's talk, when he was talking about control, of morphogen gradients, here it's not going to be that dependent on those type of kinetic parameters, but yes, very much on the polarity of these cells. Where do they decide to locate their transporters? How much transporters do they locate there in relation, for example, apical to lateral expression? Okay, so it's a very different level at which the biological control is operating to give you spatial scales. Okay, so back to the idea of foraging, and I'm not going to go too much in it, but just to say that we also have models which are like, we call them metamere models. So we've reduced even the complexity even further of the root. We just think about it as a strand of cells. This is the whole cut of the root. And we try to offer it low nitrogen, high nitrogen, low nitrogen. And we try to understand how by inverting the flux of auxin, you can actually get, for example, higher amount of lateral roots or lower at other regions. But this does work. But in the end of the day, it begs a question. And in all of the stuff I've been showing you, the polarity that we put into the models, we force it upon the model as we see it in the experiment, okay? Elliot has been showing also that in the system that he's looking at, he can get them self-organizing in relation to the mechanics, for example. But this case is a little bit different. And so we were asking ourselves, well, so we have answered lots of questions, right? But in some ways, we feel we are as confused as ever. Okay, but this is now my belief. But we believe we are confused on a higher level and about more important things. Because now the question is, how do you get this polarization? How do these tissues know how to orientate their pins, right, so robustly, and to regulate that? So this is really where my actual talk starts in that sense, because I would like to talk about the coordination of polarity, right, of cell polarity over tissues. And this is something which happens in the fly. It also happens, so these are the pins, in plants, so which are all located here facing the right, and this is the emergence of a little leaf, a leaf bud. But also, for example, you can think about, polarity over tissue. Also, for example, these are vectors are the movement of cells, right, during development. And the way we tackled and started getting insights in this was actually then not thinking anymore about multicellularity, but going back to a system which is one cell, okay? And this is the keratocyte, this is lamellipodia, so this is the part where actin is actually pushing against this front, and it's sliding over a 2D plate. Okay, in the back here you have the nucleus and you have the retracting end. Okay, so you have a polarized structure, and it can move for hours in this very beautiful gliding manner, go up gradients and so forth. So a lot is known about the keratocyte. It's a beautiful model system, which is why we started studying it from a modeling point of view. Now, behind the scenes, what is located in the front and the back, so what is steering this guy, what is telling it this is the lamellipodia and this is the back, are small G proteins. Okay, so we have these 3 small G proteins that we took from the literature, and we saw that they were interacting in this fashion here. So CDC42, RAC, and Rho. This might be familiar for some of, in some of your work as well. So in this particular model, we assume that CDC42, or this is what the literature had been telling us, and Rho, they mutually, they're antagonistic, while CDC42 promotes RAC, which promotes Rho. Now, downstream of this, we have the things that they do with the cytoskeleton. So this is why CDC42 and RAC, they end up determining the front of the cell, because they will promote branching of actin, uncapping, and so forth. So you will have more barbed ends at this region where these are high. And the back, so Rho, is actually promoting the contraction through myosin. Okay, so that's sort of how it goes through. But we asked a more basic question. Okay, so if this is the front and this is the back, how can we understand that this polarity will arise? How do we get the symmetry breaking of these proteins? Okay, so this is the model again that I just showed you. We just assume here that their interactions is going through the suppression or the activation of the GEFs, which is what is switching the states between active and inactive. So you have CDC42 GDP or GTP bonds, so the active and inactive form, and this is done through GEFs, that activation and the inactivation through gaps, right? So this is the switch. Let me know if I'm not clear here. And from here, what we do is basically we write a differential model, right? Ordinary differential model for this, actually a partial differential model here, because we're going to include also diffusion of these substances. So this is nothing else than just taking this scheme that we like to draw, right, as biologists, and reinterpreting this in mathematical terms. So here we see that this is the amount of CDC42, of RAC and Rho, the active forms. And we assume in this system that active is becoming inactive or vice versa, but they're not being created or destroyed. So it's conserved. So that's what I'm writing here. So for the inactive terms, it's just the negative of that. So this rate is the rate of deactivation of all of them, and this is just a diffusion. And so what you see here is this is the activation term of CDC42, of RAC, and of Rho. And what you see here is, for example, CDC42 is being inhibited by Rho. Rho is being inhibited by CDC42, and RAC is being activated by CDC42, right? So this is this type of scheme. Now, for the referee eye, you will see there's a little n here, and that's the Hill coefficient. And that will tell you sort of the response. So how does this inactivation, how stepwise is it, okay? And it's important to notice, because I'm going to talk about the importance of that for polarity or not. So the first time we were thinking about polarity, we thought, well, we need to have a bistable system. We need to have a state where either the frontness is low, or the frontness is high, right? Because that's happening in a cell. You have one region where it's going to be low and one region where it's going to be high. So with a Hill coefficient, which is like n equals 3, which is quite low, what we see for CDC42, the active amount, whatever you do with the external stimulus, you smoothly have it rise, but you don't get a bistable regime. Now, with a Hill coefficient of 6, which is a little bit contrived because where do you get that, but well. Then you can start getting bistability. So here, for these regions, for these values of the basal activation rate of CDC42, you can see you can get a low equilibrium or a high equilibrium. And this is the unstable equilibrium here in the middle. Okay? So we thought, okay, so that's it. This system can present bistability, so it's solved. But that was wrong. Okay, so if you would run these simulations, and what actually bistability will tell you is that some cells will be either all high and some cells all low. It doesn't mean immediately that you're going to get a cell being partly high or partly low. Okay, so for that, and you can see it here. So these are just solving those partial differential equations. So this is space and this is time. And what you see is like, well, for Hill coefficient n equals 3, you're not expecting any symmetry breaking anyway. For n equals 6, you are. So this is high, this is low. But over time, you see this wave of CDC42 highness. It takes over the whole cell. Okay, so this cell is not polarized in that sense. So what is happening here? So because of that, we started inventing a way. So it's a tool. It's a mathematical tool. And I would like to talk much more about it. But I just want to give you the taste. It's called the local perturbation analysis. And what we basically do here is we say, OK, we want to probe any type of protein interaction network and ask the question, would this be able to generate polarity or not? But we have to assume, for this thing to work, a limit. We have to assume that because the active small g protein, it is in the membrane. It's going to have a somewhat slow diffusion, and that's true. The inactive form of these small g proteins, they can be released into the cytosol through something with the GDIs. So they will diffuse quicker. So mathematically, for a moment, we're just going to imagine that the active form is not diffusing at all, okay? And the inactive form is diffusing infinitely quickly. So it will be like a parameter. And then we can ask, will you be able to get this polarity arising? So I won't explain exactly the mathematics just because of the time. But basically, by applying these assumptions, we can get a different type of bifurcation now. And this is giving you, so the black was what we had before. But now we see that there's a regime here where it is unstable. And this, please note, is for n equals 3. So we don't have to crank up the Hill coefficient now. So this is really great. So we will get polarity for free at low Hill coefficients. While this gray line here, this gray branch, is telling you what happens at a local patch of the membrane. So now what happens when I change this parameter, IC, at low amounts, you see that the whole cell will only have one equilibrium for CDC42, so it's all going to be even, homogeneous, no polarity. At a certain moment, and this is around here, you can perturb your system. That means you have to give a sufficiently high perturbation at a certain location, and that will go to a local equilibrium. So this is now polarizable, but you have to trigger it. If you crank this value even further, you're going to have a regime where the cell will be inherently unstable. So it will not be able to have a rest state. It will want to polarize, and that's what's going to happen here, right? So then you're going to get one part of the membrane being in a high state or in a low state. Just to show you that this is, this is correct. So this is what you see in the simulation. So this is a 2D cell where we just simulate those conditions. And what you see here in red are high amounts of RAC and here low amounts of RAC. So you get this polarization. And what's also very nice is that it doesn't sweep over the whole cell. And these are with actual diffusion rates as found in the literature. Because as there's more region which is becoming high in the active amount, it's taking up from the pool of inactive form, and that will then stop the wave from continuing. And that's a phenomenon called wave pinning. Okay. So now that we talked about that, can we do some fun things? So now it'll be more fun to talk. There will be less equations. So we thought, well, what about taking now the circuit, these partial differential equations of your small g proteins and throwing this in an in silico cell and see what they can do? So we did this by putting it into something called the cellular Potts model. So this is a Hamiltonian-based approach of modeling cells. It came from the idea. If you guys ever know the Steinberg, he proposed that cells could sort out due to differential adhesion, right? And what we just do in the simulation, we just take experimental measurements of surface tension of different tissues, so limb bud, pigment, epithelium, heart, liver, and neural retina. So this has been done experimentally. In this experiment, they show that the tissue with the smallest surface tension, for example, here in red, the pigment, the epithelium, it's going to engulf the one with the higher surface tension, right? And this is because of the adhesion relations. So if we adjust the adhesion of our model to be able to give these same surface tensions, what we see is, and this is the nice thing about simulation, you can throw all these cells in simultaneously, and you'll get that same hierarchical order. So the green inside, red, yellow, blue, and orange, right? And this is the simulation of the cellular POTS model, in which we're modeling cells as having interaction through adhesion. They will have area conservation. And you can appreciate here that there's all this little trembling going around, and that's the noise, the temperature of your the temperature here of your Monte Carlo simulation. We interpret this biologically as the activity of the cytoskeleton, okay? And that's necessary for the cells basically to find themselves, right? To go to this configuration. But in this case, so this is the classical cellular POTS model, cells they're not polarized, right? They're wobbling, they have these properties, but there's nothing which is giving them a front or a back. So what we did is now we couple to that our first part of the talk I was telling you about the small g proteins. So we basically allow these reaction diffusion equations to run within a cell, right? These cells are they have a very small precision, so there are many, many here pixels, so on the order of, these are really like small hexagons, right? And the reason I have them as hexagons is because we will have, for every direction of the hexagon, we will be able to look at the density of filaments, of actin filaments which are growing there. So now we have in this model actin, so small g proteins, actin, and downstream of that we will say, okay, the forces on the edge of the cell will depend on the density of pushing barbed ends, right? And the retraction of that cell will depend on the levels of rho and the myosin retraction, okay? But there's nothing specified in this model telling it, or what is the front and the back. So now let's see how it works. Oh, sorry. So if we initialize a homogeneous state, cell is going to spontaneously polarize. It creates its own front and its back, because we have these equations describing how actin the barbed end is happening, right? The ARP2-3, the capping and all that things behind the scenes. We get effectively a force going forward and a retracting backwards. So we get a gliding keratinocyte. So all the details can be found in these papers here. Now that was really cool, right? So we were very happy. But we started seeing that the model had some problems. And one of the problems was if that cell was allowed to run into a wall. Okay, so it has its front, it has its back, it's just gliding, and then it's running into an obstruction. And this is what you're going to see in this frame here. And that happened at a second moment that we were having these discussions with Leah Keshet, because she was also very interested in what was happening with the phosphonocytides. So this is another level below the small g proteins and the cytoskeleton. And initially we thought, well, phosphonocytides aren't important at all for polarity, because we can get all the symmetry breaking without them. And now I just want to show you here in this simulation why the phosphonocytides are important, so this other level. So this cell here is just with the small g proteins. This one has the phosphonocytides, and this one has them too strongly acting on the system. And so this green line here is the wall. So what you see is for the normal only small g protein case, when the cell collides against the wall here, it's going to actually break in two, because that front is starting to get split, right? It's changing its shape, and it can't, on a faster time scale, resolve itself. This one here actually gets stuck, because it has a very localized front, this hot point, and it flanks its myosin tails around it and just gets stuck at that wall. This one here, though, so with intermediate dosage of this interaction of these phosphonocytides, it's actually having a front which is very difficult to separate and will randomly choose a direction, up or down. Okay. So these were the type of insights we were getting in relation to the biology. So because now we can see that with phosphonocytides, let me just pause it for a moment. Cells can resolve very well conflicts. We just started the simulation where we randomly have a confluent configuration with many, many cells. They're just different colors here. But they're all moving because they're all polarizable. And what you see here is after some time, and these colors are telling you the direction in which cells are moving in. In the beginning, you saw they were just randomly moving all directions. They start making microcurrents. And in the end, they will all flow in just one single direction. Now, what's important to realize in the simulation, at least what we learned from the simulation, is that there is nothing in this field telling cells to coordinate. Cells do not know the direction their neighbor is going into. It's not that they know the velocity vector or what their neighbor is doing. It's just because they are initially ramming into each other, you can see here, and due to that changing their shape, that their polarity will reorientate, so you can see these swirly patterns, and you will end up getting this, this very nice tissue-level collective behavior and polarization in movement. Okay. So by adding lots of these polarities together, you start getting these emergent coordinations. Okay, so what does this have to do with the plants, right? It seems like totally a different planet, right? So when we look at the leaf epidermis now, we look at that top layer, we also see that cells, over the development of the leaf, they can grow. Some of them grow and continue being brick-like shape, having like this polar axis. But many of them, and these are called pavement cells, they will become these very funny shapes. They look like, can you see them well here? They look like little jigsaw puzzles. So instead of having just one front, one or two, whatever, right, they can have up to 42 fronts. They will become more and more and more and more complex. They're just going to grow out these sort of ramified branches. And the way this happens over the development of the leaf, so this is the leaf, is that they begin at the base of the leaf very isotropic, so very like squarish or hexagonal. And as the leaf grows, there's like this wave of differentiation that cells become pavement-shaped, right, like a jigsaw puzzle shaped, and in the end, all of them will have undergone this differentiation. Okay. Now, what is behind the scenes here are, again, small g proteins, right? And so that's why it's so amazing, right, to look at these different systems and realize that what's controlling here, these processes are very similar. So again, we have small g proteins, so they're called rows of plants, ROPS, okay? They're being activated through the GEFs, where then they can do their downstream effects, as we saw before for the animal cells on the cytoskeleton, like actin and microtubules. They can be deactivated then by GAPs, right? So now they're in this inactive off state, and where they can be internalized through GDIs, and then they can diffuse faster. So we have exactly all the same elements: the switch, the on and off, the slower diffusion, and the higher diffusion. So mathematically, it's actually equivalent to the system we saw before. But it raises a question: why does the previous system just give you one front and one back, and why can we get in such a system so many fronts? Just to show you that this is, well, it's happening in the pavement cell through, you can see here the downstream effectors, but also, for example, on the root. So this is a very big cell. Prior to these root hairs emerging, you see these polar localizations here, clusters of ROP2. So ROP is very important for that outgrowth. So I just will repeat the logic of what we did for the animal cell. I won't get too much in the details, because the objective of this talk is not to talk about the network. But again, we ask the question, so how can you get these different states coexisting? One of the indentation state, which is given by ROP6, because ROP6 is high where you have the indentation, and one which is the protrusive lobe state, which is the ROP2, which promotes actin branching. So here we have, this would be our CDC42, and this would be our row, right? ROP2 and ROP6. So again, we apply the local perturbation analysis because we know we have a slower diffusion species and a higher diffusing species. We have an interaction network, which is different than the other one, but we can again apply the same strategy. We ask, can we get lobe and indentation coexisting? So these would be the first, the reaction part of our equation. We can, through a quasi-steady state approach, we can just reduce it to two equations of ROP2 and ROP6. We apply the spatially explicit analysis, right? So these are the diffusions, and the local perturbation analysis. And then we get exactly that atlas that I showed you before. So this atlas will tell you, now with this bifurcation parameter being Auxin, because Auxin is, if I forgot to mention, it's activating both of those pathways. And what we see here is that at low Auxin regimes, your cells should not be able to polarize. At higher Auxin regimes, you are able to polarize only if you give a big enough trigger to it. So here it doesn't polarize, and here it does. So this is in this region. At a certain moment, Auxin will be high enough that the cell will polarize spontaneously. And this is what happens down here. And what you can also appreciate is now we get some type of like Turing bifurcation, where also the frequency here of how many lobes do you end up with depends on the Auxin amount, or on the activation amount of this network. Okay. And so just to show you, Zhenbiao Yang, he did these experiments where with different amounts of Auxins, he could also see that he could get different amount of lobes being formed with somewhat optimal around this dosage here. Okay. So now, let's try to simplify everything I'm talking about, right? Because CDC42, RAC, RO, ROP, there's a lot of names. And we started thinking, what is the minimum that you need to polarize a cell? I mean, all cells seem to be able to polarize. For every system you're going to look at, there are going to be other players. But what would you need, in principle, to have a cell have a front and a back, right? Something like a keratinocyte or a pavement cell. And so what we realized, and this is a work with Rico, Chemik, Stan, and Katie, and there were other people involved as well, but these are the ones I most discussed with. So what we realized is you just need to have two. Well, actually, with one species, you can get already the break of symmetry, but to have a robust polar cell, you would have to have two, let's say, small g proteins, or two type of proteins, A and B, which can have the on form, which is A star, B star, which will be in the membrane, right? And they will have a off form, so the A and the B, which can diffuse a little bit more quicker, right? And with mutual inhibition between them, you can easily get them to polarize. So the way we thought that this is giving us is that, well, this would be what we would imagine would be the most simple thing that you would need to be armed with as a single cell to be able to polarize. So what happens now if these cells would come together? So this is very sort of Gedanken experiment, right? So we take them cells, we put them together. So here they're homogeneous. They spontaneously polarize in a 1D string and in a 2D plate. Well, because there's no interaction between them, they just polarize in any random fashion, right? So this is just, that's sort of obvious. But now, if you give any type of coupling between them, in this model, we imagine that these proteins, they could touch each other, but this could happen through other complexes, of course. But if there is some type of way that A star can feel B star on the other side, and that would enhance that mutual inhibition, what you start seeing is you get alignment. So the front of one is attracting the back of the other, and so forth. And what you can see is that these alignments in 2D, so you get swirly patterns. They're not perfect. It's coordinated, but it's not that you get like from one side to another. But yet you do get these coordinations. So we thought, okay, but how do you get it so nicely coordinated from one side to another? What could be possible here? So we thought about what are the external signals you could give. And different things what you could imagine is, well, if you had a morphogen gradient, right, as we've been talking the first day, you could imagine that this morphogen gradient, or this morphogen, is a ligand. So a cell could have more in its front and its back, right? And that would do something with these complexes. Or that it would be something like a transcription factor. So one cell has more than its neighbor, and so forth. Still begging the question, how does one cell know that its neighbor has more or less of that transcription factor? But because these cells are polarized, right, very naturally, without them knowing what the other one is experiencing, you can get these perfect directions in the following ways. Either you say there's an organizer. So here, these cells are artificially all having just the A star and these just the B star. Then everything in between will very nicely follow as a sandwich in alignment, okay? Another thing you can do is then give that transcription factor gradient, in which a transcription factor is producing more of the red protein, of the A protein. Then you will also get now a polarization down the gradient. Or you could say that this is a ligand, and there's even some noise here, and that ligand is promoting the A star going onto the membrane. And then we get the polarization going up the gradient, okay? And to finalize in the last 2 minutes, how would this work then for plant tissue? Now, plant tissue doesn't have the luxury that those proteins can actually touch each other, not even with complexes, because they have this, this cell wall in between. So here we just hypothesize, because we know there's a lot of signals being pumped or excreted into the cell wall. We imagine, so what if A star now, so the front of the cell, would also be able to secrete a molecule, right, a diffusible molecule, which would inhibit A star itself. Okay, so it's excreting something which inhibits it. And interesting enough, if you apply this rule, what happens is that you start getting coordination over the tissue. And if you have like some organizers, I don't have this here, you can get very nice coordination as if it were like along a route and so forth. So in the last slides, I would just like to say that what we're doing currently is now we have an automatic platform in the lab where we're looking at mutants of the small g proteins. We're able to automatically segment all those cells. We had to invent something which we just recently published, which is a way to quantify cell shape, and it's based on elliptic Fourier analysis, but it had to be altered. So it's called the lobe contribution elliptic Fourier analysis. And we're sharing this with the community. I really recommend you guys, for any problem you have on shape analysis, to see if this would work for you guys. It's quite powerful. But this is allowing us now to quantify the level of defects we see in these shapes. But it also allows us to automatically track cells over time on the leaves, right? Which allows us now to think about models of leaf growth, how does the scaling work, why do leaves stop at a certain moment, and so forth. But most importantly is that I think these concepts here, that you can get the coordination of tissues, which is allowing information to be passed on fast timescales. This is linked, right, to these ideas of, for example, bioelectric fields, which we saw from Michael Evans' talk. It is, I wouldn't say it's similar, but this plane of polarity idea of having coordinations and having our landmarks. It rang a bell when I was listening to Jennifer's talk on the intercalation and the PAR genes. And I would also say that even thinking now, what is missing here is how would these things now link, for example, with excitable media, calcium waves, and possibly could one couple now these biochemistry networks to mechanics and to the directed transport, as we heard, for example, in Meyerowitz' talk. So for me, the frontier here is now to think about these conceptual links and try to understand them back to the molecules, right? How are these different instances, these different organisms, these different problems solving very fundamental issues of coordination over tissues. So with that, I would just like to acknowledge the people which were collaborators in the work I've been talking about today. So Stan Leah, very close by in Vancouver, Rico, Alan Champneys, and Andrew Bangham. And these are the students which were involved. Katie was a student of Rico, and all of these students were in my lab working on these projects. So they were really great. So with that, I would like to thank your attention, and hopefully there'll be a question or two.
We have time for some questions down here in the middle. Okay, Damar.
Very nice talk. Richard Smith had this paper recently that you have puzzle cells to minimize hoop stress. Can you comment on that? How does this link to your biochemistry?
Yeah, so there is another model which is proposing that the growth of the cells would create conflicts, and you would get these indentations, right? So what I would comment is we have mutants where you can have exactly the same growth, but if you, like, for example, knock out one of the effectors, you don't get the ripples. So I do think that probably that is contributing a lot, but you can still bypass that with the biochemistry.
Okay, but you would not argue that they may be coupled, that there's maybe
I totally think they're coupled. I totally think they're coupled. So, well, I didn't have time to show it, but what we're proposing is that once you start growing, you will have mechanical coupling between these cells that will change the shape, and the shape also changes then your intracellular patterning. So you have these coupled timescales. I think that the important experiments to do in the future would be to have clones of mutants and then look at what's happening between the interaction of a wild type, which has okay biochemistry, and its neighbor, which doesn't, but which are mechanically coupled. Because then you can untie a little bit what's the influence of the mechanics and the biochemistry. But certainly both are very important, but I just don't know to what extent one is, could you say one is dominating the process or, yeah.
Okay, thank you.
We have time for one more question, right there in the middle. It's coming to your other side here. There you go.
When you speak of polarity, are you referring to electrical polarity or magnetic polarity,
Okay.
or both?
Neither. So actually, when I talk about polarity, I'm sorry, I mean cell polarity in this case, which means that if you have a cell which has diffusible substances in it, it'll be homogeneous as everything is at the same concentration because of diffusion. And once it starts to get certain regions having more than others, right? So then that would be polar, right? So for example, if you would be able to, does that answer your question? So I'm not talking about electromagnetism.
Okay.
I'm talking about the distribution of proteins which have downstream effects on biological things like cytoskeleton. Okay. However, your question is very interesting because an analogy to this, what I'm saying is that every cell, because it inherently will be able to polarize, and it does polarize, you should think about it almost like a little magnet, like a dipole, right? And then when you put them together, you would be able, through interactions, to get them, getting higher scale coordination, like you would for a ferromagnetic. So that would be an analogy to this. And I can also imagine that these concepts on another level, I mean, they don't have to be small g proteins, but could also apply to bioelectric fields. But I do think that cells inherently always polarize. It's not that they're a homogeneous bag of nothing where you have to give a signal from outside for it to polarize. It intrinsically is polarizable.
Okay, sorry, we have to move on. Please find Veronica during the break. Thank you so much for that wonderful talk.
Yeah, thank you.